So hello, everyone. Thanks for coming to this CRISPR gene editing workshop. We're really going to talk about how you can teach CRISPR in your classroom. And we'll spend a little bit of time on the lab itself, but probably save the bulk of that to a webinar that we're going to have in August, where we'll be doing the hands-on lab. So what I really am hoping to do during this one is kind of get you acclimated to how you might present this in your class, and where there's some good resources, some free resources, especially that you can do with your students as well. My name is Leigh Brown. So I live in Texas and I'm a curriculum and training specialist for Bio-Rad. I also have Tamika Stubbs with me in the chat. She'll be answering questions and helping out. The two of us in Damon, we cover the whole country, basically doing trainings and helping teachers. Aaron Callis is based at home and he helps remotely. So you've probably talked to one or the other of us if you've ever gotten anything from us. We're really here to help you troubleshoot your labs or give you ideas about how to use them. We've done all these labs hundreds of times, and so we've learned a lot in that process. And so you'll get to see some of the tips and the tricks that we've learned over the past few years of doing these. So we're going to start off today by talking about what is CRISPR, talking especially about like some of the discovery. There's a lot there, it could be a whole semester's worth of conversation, but we'll kind of hit the high points. We'll talk about how bacteria use this for immunological memory, so adaptive immunity in bacteria. We'll talk about specificity with the PAM sequence, and we'll pull out that paper model. You guys all got an email with the paper model link, but it's also in this PowerPoint, and Tamika can put it in the chat as well if you want to download that. You don't need to do that for this workshop. I'll have it digitally, but if you did want to manipulate the pages or the strips of paper yourself in front of you, you're welcome to do that during the workshop. We'll do a quick overview of the lab, the CRISPR lab, and then we'll have a little bit of time for question and answer and a survey at the end. So let's start off talking about what is CRISPR. So in the chat, if you don't mind, just writing some words that come to mind when you hear the word CRISPR, or what do your students think of when you say CRISPR? Do they have any kinds of, have they heard any news stories, or are there certain words or phrases that they might think of? Yes, I'm getting gene editing, enzymes, DNA, students, chicken nuggets, me, oral exams. True, yeah, right. The CRISPR acronym was chosen because it's kind of catchy, but it also may cause a little bit of confusion. So yeah, bacteria, this is where it was certainly found. Students think of designer babies. Awesome. Yeah, and you can just keep writing your responses in there and we'll kind of collect those ideas. But I think some of this was covered. We mentioned a few of this. There's been a lot in the news lately about sickle cell disease and how we're using CRISPR to prevent the conversion of fetal hemoglobin into adult hemoglobin and prevent the symptoms of sickle cell as a result. So here's just a few images from recent headlines. Up in the top, we have the twins. I don't know if you are familiar. A Chinese researcher in 2018 kind of dropped this bombshell that he had altered a gene in human embryos that resulted in these two babies. It's not actually these two babies. Sorry, I don't have a copyright free picture of the two babies. But the twins carried a disabled HIV receptor gene. Turns out that they were kind of mosaic. So some of their cells had that and some of them didn't. And this was kind of caused an international uproar because of the bioethics concerned with this, right? And so there's a lot of good bioethical discussion you can have about this. What kind of regulations should we have? We're at the point now where we could do whole genome sequencing of people just for several $100s. That's just going to get cheaper over time. Do we look at doing that for everyone, as they're born, and figuring out what genetic diseases or issues they might have, even what medications might work better, right? So a lot of valuable information that can come with that, but and we have the potential to fix that with CRISPR, but it's fraught with peril here, right? The woolly mammoth and the passenger pigeon, those are both de-extinction projects. And George Church's company, Colossal, their goal is to bring back the woolly mammoth by using CRISPR to genetically edit the DNA of Asian elephants, kind of their relatives or cousins, I guess you should say, And the hope is that these woolly mammoths will be more cold adapted. They can put them in the Arctic where they'll kind of trample down trees and the snow and restore the Arctic tundra. Why do you want to do that? Well, the Arctic tundra will prevent more. So as the permafrost melts, we're releasing more greenhouse gases, right? And so bringing back that tundra will prevent a lot of that. The grasslands will reflect more sunlight, and we'll get rid of that insulating snow, and that means that the ground will stay frozen instead of thawing, right? Stopping, in crops, we're using CRISPR in all kinds of ways. So stopping the browning of bananas, that's next step. USDA just approved that, I think, last year. I haven't seen it commercially available yet, but that's the plan. And we're already have commercially available CRISPR mushrooms. Again, a lot of good conversation here that you can have with your students about USDA regulations on this. So since CRISPR tends to not introduce DNA from another organism necessarily, you can just kind of upregulate or downregulate the genes that are already existing. Then they've deemed that the CRISPR mushrooms, which have a enzyme block that helps prevent browning, that those are not actually genetically modified. So that's kind of an interesting twist on, GMO crops and things like that. I suspect that we'll see a lot more CRISPR coming into play here. Mosquitoes, of course, we're looking to get rid of breeding populations of mosquitoes. There's lots of different trials with this out in the news. I won't spend time here. Damon has a good talk on some of these. I think Tamika has the link, or she'll put the link in the chat for that as well. We have a whole YouTube playlist on different CRISPR applications and kind of this talk as well. And finally, sickle cell anemia. So again, we have a whole talk on this, but we're using CRISPR. Vertex is one of the companies involved in this, and they're using CRISPR to edit fetal hemoglobin so that it doesn't convert to adult hemoglobin. Fetal hemoglobin is not sickled, and so it prevents sickle cell disease. So really exciting stuff here. But the big picture is CRISPR is a way that we can edit genes in a way that's really targeted. So we can hone in on exactly what we want to change, and very precise, so without a lot of off-target effects, and relatively easy to use. So this is a lab that students can do in a classroom, maybe not with humans, but with bacteria, so they can see how that whole system works. So this got the Nobel Prize in 2020. Jennifer Doudna and Emmanuel Charpentier were the researchers who built on a whole legacy of other science out there, right? So we'll walk through some of that science, but they were really the first ones to show that we could use CRISPR, which was found in bacteria, as a way to edit human genes. And so that was really pretty groundbreaking, right? So what is CRISPR? Way back in 1987, the last century, it seems like, for me, that seems like 20 years ago, but that's, not. But 1987, Ishino at Osaka University in Japan discovered these short repeated sequences in E. coli. And I've got the papers linked in the notes to this PowerPoint. So if you want to look at those, you're welcome to do that. So that region of DNA had these short repeating segments of short repeating sequences that each had these identical 29 base pair sequences, and they were interspersed with a 32 base pair sequence. Okay. Now, the acronym CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. So all of these repeats were in a cluster, right? So later we refer to that as the CRISPR array. They were regularly interspaced by those seemingly random sequences, about 32 nucleotides long. They're palindromic, so the repeats read the same way going forwards and backwards, okay? And there are several of these, 5 or 6, right? Later on in the 90s, Francisco Mujica in Spain was working on his thesis on halophiles, these salt-loving archaeobacteria. And when he sequenced the DNA, he also noticed these repeats. Searched the literature, right? We didn't have, it wasn't as easy back then just to go in GenBank and type in repeats or whatever. So it was pretty painstakingly slow, but he did find that previous discovery by the Japanese group in E. coli. And since he found these repeats in archaeobacteria as well as in E. coli, and later we find it in a lot of other bacteria, he figured this must be pretty important, right? So later in 2001, he coined the name CRISPR with another scientist, and characterize what we now call this CRISPR locus, which is these regularly repeated sequences of DNA with those spacers in between. Now, the spacers weren't at first the subject of all the research, right? Those were just thought to be kind of random DNA interspersed between the repeated elements. But in 2005, Mojica was sequencing all these, these CRISPR arrays and found that those spacers, the DNA there matched DNA from bacteriophages. So that made him think, and he, his hypothesis there that CRISPR was some sort of adaptive immune system. So they discovered, he and his lab discovered that viruses can't infect if they have that homologous spacer sequence in their CRISPR array. So that's what kind of led him to hypothesize that maybe this was some sort of immune system for bacteria, right? And it turns out that's exactly what it is. So the spacers match the DNA from individual bacteriophages. Whoops, hold on, I think I went too far here. There we go. So the whole CRISPR array, this region in the bacterial chromosome, and remember this is part of the bacterial chromosome, it's not on a plasmid or anything like that, consists of these repeats with the spacers. Remember, those come from the bacteriophages, along with CRISPR associated genes. So you'll see Cas, that's for CRISPR associated. Cas9 is a protein from one of those, right? And there's also proteins that are helicases and other nucleases that cut DNA in that locus, okay? So let's take a little bit of a closer look at that idea of the bacterial adaptive immunity. And this was a big surprise, right? For most of, previous to this, we thought that bacteria had some kind of non-specific defense mechanism against bacteriophages. Restriction enzymes are one of those defenses, right? But having an immunological memory wasn't really something that we considered, okay? So in 2007, two French food scientists were working at Danesco, which is a yogurt company in France, and they were looking at Streptococcus thermophilus and realized that bacteria was using CRISPR-Cas9 to defend against different bacteriophages. They actually wound up exposing different bacteriophages to those bacteria in order to make them more and more resistant. Okay, so let's look at how that works. So bacteria are always under attack from viruses called bacteriophages, and you can see one right up here in the corner, right here, right? And whoops. When bacteriophages infect bacteria, they inject their DNA into the cell, and then they use that bacterial machinery to replicate, making a ton of virus particles and eventually killing the cell when they burst out. So obviously bacteria are not, big fans of this. It kills them. So they want to prevent against this. So let's break down how the CRISPR system works for providing immunity to bacteria. So the system uses these Cas nucleases, this is Cas1 and Cas2, to snip out a little segment of the viral DNA. And that piece of viral DNA is known as the protospacer. We'll come back to that in a little bit. And then the Cas proteins insert that segment of viral DNA into the CRISPR array as a new spacer, okay? And so we're just taking a snippet of the viral DNA and sticking it directly into the CRISPR array, right? And so here we have our spacers, and that spacer is going to be next to the spacers from other viruses that previously infected the cell. So now this bacteria has memory, right? Immunological memory here. The spacers are between the CRISPR repeats, and this kind of vaccinates the bacteria. So each of those spacers is a history of a previous infection. The CRISPR array is transcribed into one long piece of RNA by RNA polymerase, and that long piece of RNA is called the pre-CRISPR RNA, pre-CRRNA, you'll see that. Together, another RNA binds with that pre-CRISPR RNA, and they associate with Cas9, and then there's an enzyme that will cut that pre-CRISPR RNA into individual pieces, so they each complex with that Cas9 and the RNAs have the spacer from one virus, okay? So at the bottom here, you can see that there are different Cas9 CRISPR tracer RNA complexes here, and each one of these is specific to one of these viruses, okay? Now, that is how we build that memory, right? How does that help us later? Well, if the same bacteriophage comes along and infects the cell, Cas9 will work in conjunction with that guide RNA. So the CRISPR RNA and the tracer RNA together are called the guide RNA, right? So Cas9 works in conjunction with that guide RNA to scan the DNA and target that same sequence in the viral DNA. So once it finds a match there, Cas9 is a nuclease and it will create a double-stranded break, okay? Once the virus, or once that DNA has a double-strand break, the virus can't replicate anymore. Viruses don't have a way to repair their own DNA, right? So that double-stranded break incapacitates the virus, and now that virus can't replicate, okay? Any questions on this before I move on? Let me check the chat and make sure I'm not missing anyone. Okay, all right, great. Looks like we're pretty good. Yeah, feel free to either unmute yourself or post in the chat if you have any questions at any point. Okay, so now let's look at something called the PAM sequence. If you're looking at that's a little bit closer up. So on the left, we have that CRISPR array right here. And on the right, we have what's going on with the CRISPR-Cas9 system targeting the viral DNA, okay? So our target sequence is, the sequence in the virus. But that sequence is also in our CRISPR array, right? So that's the same sequence. That DNA is the same sequence. It came from the virus originally, tucked it into the CRISPR array, and now we're trying to target it in the virus when it affects again. How do we keep Cas9 from cutting that spacer sequence when it's in the CRISPR array? We don't want to do that, right? So these are all the same sequence. So Cas9 needs to cut the viral DNA, and other Cas proteins are going to cut the incoming infection, right? The DNA as it comes in. We want to cut that viral DNA, but we do not want to cut the same DNA when it's in that CRISPR array. And the reason is because double-strand breaks in bacteria are typically lethal, right? We'll talk about some repair pathways, but oftentimes these are lethal. So bacteria do not want to cut that spacer DNA when it's inside the chromosome. So how does it keep from doing that? So if you have your paper model, you can go ahead and look at this, or you can just look at the digital one I have here. This is from, and I think Tamika might have put the paper model in the chat already. It's in the student guide of the, it's in the student manual, so it's at the back of that. There's also a separate PDF, is the one I emailed, that has just the paper model stuff pulled out. I like to show this to students just to kind of get them thinking about what the PAM sequence is. So we're looking at a strip of DNA here, and the PAM sequences are highlighted in green. So I want you to look at this and then let me know, what do you notice about these PAM sequences? What kind of pattern are you seeing? And go ahead and write that in the chat if you don't mind. Okay. Chat. All right, I'm seeing a lot of GGs, G-Rich, GGX, yep. 3 bases, 2 Gs followed by another base. You got it. Yeah, you guys are on it. So exactly. So what we're looking at here is the sequence 5' NGG to 3'. N stands for any So it might be G, A, T, or C, right? So you can see that we see, I'm just going to show you up here on this one, we have 5' AGG, CGG. Now, typically we're looking at 3 nucleotides. I think in different bacteria, this might be different. And in different bacteria, the sequence might be different. This is in Streptococcus, though. So this NGG sequences. Wherever you have GGG, you'll have 2 PAM sequences overlapping. So that's why you see these right here. So we have a GGG, which is 1 PAM sequence, but we also have overlapping with that TGG. So that's why we have 4 nucleotides highlighted there. Okay, so those are the PAM sequences. Okay, so how does that help us? Well, when Cas1 and Cas2, actually, let me go back one. I'm going to show you this right here. So this DNA in the CRISPR array is called a spacer. When it's still in the viral DNA, right, as it's the bacteria affected it into the bacteria, when it's still in there, it's called the protospacer. So before it becomes a spacer, it's a protospacer, right? And next to that, we are looking for this sequence, and that is where the PAM sequence gets its name. It's for protospacer adjacent motif. So the sequence next to the protospacer. Okay, makes sense. We already decided it was NGG, so that's what that looks like. And that's what Cas1 and Cas2 will cut out a region of DNA upstream from that. Whenever they see, if they see that NGG, cut out a region upstream from that and put it into the CRISPR array. Now, that same sequence, that PAM sequence, also needs to be present in incoming bacteriophage DNA, right? So if this is our target DNA here, right? The PAM sequence is highlighted in green right here. It might be kind of hard for you to see. So that PAM sequence is also going to be NGG, right? And it could be a different one. It could be AGG in one and CGG in the other. That's fine. It's just NGG. That's what we need. Okay, so the PAM sequence is present in the viral DNA or in our target sequence, right? Now, think about this. What do you think, what nucleotides do you think follow the spacer? And this is really in the repeat area, right? What CRISPR's named for, that repeated region. What nucleotides do you think are in that start out that spacer? So that start out that repeat. So we have the spacer here, and then we have 3 nucleotides. What nucleotides do you think those are? And I want to remind you that we do not want our Cas9 to cut here, right? So what do you think these nucleotides would need to be? It's a little bit of a trick question, I guess.
Mary says not NGG, Julie not NCC, Ashley XXG.
Yep. Basically anything other than NGG, right? So we've got, in this case, GTT. And again, this is going to change from bacterial system to bacterial system. This is what it is in Streptococcus, but GTT, it's not NGG. And so that will prevent Cas9 in that guide RNA from cutting the spacer in the bacterial chromosome, right? So hopefully that makes sense why the PAM sequence is important. We need, for everything to work, we need that target DNA with a PAM sequence, and we need the Cas9 and the guide RNA to make the cut, okay? So that GTT, or the not NGG, is present at the first 3 nucleotides of each of those repeats. Okay. Now let's take a little bit of a closer look at the CRISPR RNA and the tracer RNA. So the CRISPR RNA, remember, is cut from that long CRISPR array, and it's cleaved by an enzyme and just an RNase. Okay, so an enzyme that cuts RNA. And now we have that 20 nucleotide guiding region. This is from the viral DNA itself, right? This is the spacer DNA, right? The 20 nucleotide guiding region that will target Cas9 to a match in the target DNA when a new virus comes in. It also associates with this tracer RNA. Okay, so this is the CRISPR RNA, and this is the tracer RNA. And the tracer RNA is just kind of providing some scaffolding It also helps target that CRISPR RNA to the Cas9, but mainly some scaffolding. So some of the work that Dwalden and Charpentier did was combining those two into a single guide RNA and really realizing that we could create these ourselves, right? We can order, they're now commercially available. You can just order these, and the 20 nucleotide guiding region you can create to target whatever gene or region of a chromosome or a genome that you want. So you have a 20 nucleotide basically recognition sequence now. It's also got that scaffolding region, so those hairpin loops. And that was part of their Nobel Prize winning work, the single guide RNA, and using that to target DNA in human cells outside the body, right? And so that's what got the Nobel Prize in 2020. They published, I believe, in 2018, is that right? I'm gonna have to look at my notes now. So just kind of to wrap everything up, big picture now, we've got Cas9. Uh-oh, I've got to hold on, sorry. I've got a mute situation. Okay. We've got Cas9 and Cas9's a nuclease. It complexes with a single guide RNA. So now we're kind of looking away from this bacterial system and how we're using it in research and hopefully to fix some of these situations that we talked about earlier. So Cas9 with a single guide RNA, that single guide RNA has a guiding region that scientists have designed to target whatever gene they're interested in. And the single guide RNA provides kind of the map for Cas9 so it knows where to go and cut, okay? And so together, those two will target DNA and make a double-stranded break. So let's take a look at how that actually happens, and then we'll go to your DNA model. So here's our target sequence here. This could be, maybe you're looking at the gene for cystic fibrosis, for example, right? And we have our Cas9 single guide RNA complex. And that single guide RNA, that guiding region might be some location in the cystic fibrosis gene, right? So there's kind of the whole thing. And the DNA is kind of being unwound as it goes. Wherever there's a PAM sequence, the DNA will be checked to see if there's a match between the guide RNA and the target sequence. And so when there's a match, Cas9 will form a double-strand break, will cut all the way through that DNA, 3 nucleotides upstream from the PAM sequence, okay? And so we get something that looks like that. Now, like I said, double-strand breaks are lethal, and so cells have different repair mechanisms. So let's look at a couple of those here. 2 main pathways, non-homologous end joining, where you have your double-strand break and nucleotides are just kind of inserted or taken away willy-nilly. And as you might imagine, this causes a lot of frame shift mutations. So this tends to not be a great fix, right? Oftentimes it's lethal. You have disrupted DNA either way, okay? The other method that bacteria use, or cells use in general, is homology-directed repair. And homology-directed repair uses a donor DNA, a piece of donor DNA. This is something that scientists could put in with their CRISPR-Cas9 system into the cell, or, when it happens naturally, it's, coming from the other chromosome, for example, right? So this donor DNA is there that's not, edited, for example, or is the correct version of the gene, maybe, right? And that donor DNA can be stitched into that insert using different DNA repair proteins, okay? And so once we add that donor DNA into the cut DNA, now that DNA is repaired, right? So there's kind of the big picture. We have Cas9 and a single guide RNA working together to target and cut the DNA. And then once that DNA is cut, we can add a donor DNA template, right? Or piece of donor DNA that has the change that we're trying to make. And DNA repair proteins will stitch that template into our cut DNA. Okay. Any questions on that? We're going to go ahead and do the paper modeling activity now. So if you want to get out your paper model, you can do that now, or you can do it after we're done with this. Either way, I'm just going to show it to you digitally because that's easier than trying to look at the camera on my desktop over here. But the CRISPR Cas9 paper model is in the student guide. You can download that for free. You don't need to get the kit, to look through this. And there's a sheet that has a bunch of different DNA strips and 2 of these single guide RNAs. There's another sheet that has Cas9 on a full sheet of paper. The intent there is for you to leave Cas9 on the full sheet of paper so you don't have to cut around all those squiggly lines, and then the DNA strips you'll want to cut out pretty close to the line. Paper trimmer works great, and a single guide RNA as well. So we made the line straight so you could cut it out pretty easily. That's kind of how you start, right? So there's the PAM sequence. This is when you could PAM sequence with your students if you wanted, or you could, front load it like I just did here. And the first step is to add that single guide RNA and put it right on top of that guide RNA binding area on your Cas9 protein. So something like, whoops, hold on. That. Okay, and we're looking at, there's 4 parts to this, so we're looking at part 1. So you've got single guide RNA right like that. And you'll notice that the Cas9 has an indication where it cuts and also where the PAM site is recognized. Okay. So the next thing is you're going to take one of those pieces of DNA. We're starting with DNA strip 1, and you'll note that it can go, that the letters, one of them, the top half is upside down, right? We've actually flipped this whole strip upside down in this case. It's just so your students can more easily match the DNA so they can read it a little bit easier. So that's the only reason it's like that. Of course, Cas9 will scan both ways on this strip of DNA, on this region of DNA. And every time it encounters a PAM site, we can stop there and look and see if we have a match with our guiding region on the single guide RNA. So let's go ahead and do that here. Now, I didn't stop at the first 2 because it doesn't give us enough DNA there to make a match with a single guide RNA, right? It would only be a few nucleotides. So I'm stopping at this first one right here to see if there's a match. So we want to look for a match between our guiding region here and our DNA sequence. And we do not get a match, right? G and A, C and U, this is all wrong. Okay, so let's slide down a little bit to the next one. And do we get a match here? A, G, U, C, U, no good. Okay, let's slide down again. So we have, again, no match. Okay, so one more time. And now let's look. So we have TA, AU, AU, CG. Aha, this looks like it might be a match, right? And if you look, these are complementary all the way down. So our guiding RNA, our guiding region of the single guide RNA matches our DNA here, okay? And if you look up here, this sequence on our single guide RNA matches that complementary strand up here, right? Except there's U's instead of T's. Okay. So that's kind of how they are scanning the DNA. So the modeling here is having CRISPR-Cas9 scan DNA and find where that target site is. Once they found that, they could just draw a line right here down the 3 nucleotides from the PAM sequence, upstream from the PAM sequence, right? Draw a line like that and use scissors and physically cut that DNA strand apart. Okay. So that is how you cleave target DNA with this paper model. Any questions on that? I don't know if anyone's doing it at home or not. If you are, let me know.
Leigh, there is a question in the chat from Anna.
Yep.
I don't know if you want to look in or like me to read it to you.
Go ahead and read if you don't mind.
Oh, sure. So bacteria read PAM on one strand and viral DNA on the other.
Oh, wait, say that again. Hold on. I'm sorry.
Sure. So it's kind of like the inflection that we have to go with, right? So bacteria read PAM on one strand and viral DNA on the other.
So it gets a little bit confusing. So what we're doing now is we're kind of moving away from the whole viral bacterial immunity system, and we're just looking at how scientists are using CRISPR-Cas9 to target DNA that they're interested in. So this strip of DNA, I mean, it could be viral DNA, but let's think of it more like a DNA, a piece of DNA that you're interested in targeting. Like I said, cystic fibrosis gene, for just to use an example here, right? And so this DNA would come from, you know, mammalian cells, for example, or you know, whatever you're trying to target. In our actual hands-on lab, we are looking at bacteria, but we're actually targeting a gene in bacteria, a LAG-Z gene. So it would be the LAG-Z gene in that case, right? And so this DNA has the PAM sequences in it. The viral DNA, when we're talking back at the adaptive immunity portion, right, that also has PAM sequences too. These just pop up quite frequently in GG. It's going to happen pretty often. But every time Cas9 kind of runs across that, it will stop and check for a match right there. Okay, so it doesn't check for a match everywhere, just wherever there's a PAM site, it will check to see if there's a match between that target DNA and the guiding region of the single guide RNA. Did that make sense? Or did it clear it up?
I'm sorry, I was just trying to put it together. To me, what it looks like is like it's reading the sense string or the antisense strain. So when you actually construct this, you have to have the PAM sequence upstream on the other side of the strand. Does that make sense?
So you're right. And I have there's a capstone project that goes into how to build these and how to do a blast search. So you can do some bioinformatics with this, but you're exactly right. The important thing to remember here is that enzymes aren't like, there's not a top and a bottom, right? They're going to scan, Cas9 is going to scan both sides. And so there can be PAM sequences on both sides, and you can cut on the sense or the antisense strand. Either way, it's going to be a double-stranded break with this system. There are other Cas nucleases that make a single strand break or cut somewhere different, right? So there's different things that you can do, but Cas9 is the most well-characterized, the most well-studied at this point. But yes, you're exactly right. When you're looking for, you know, if you're designing a guide RNA, which I'm about to get to, you'd want to find, you'd have to figure out that PAM location first and make sure that there's a PAM location and that you're cutting somewhere that you could fix your gene near that, right? Hopefully that makes sense. And let me know if you have any other questions or if I answer that sufficiently. Okay, so, and it's a good segue to the next part 2 of this DNA model. The next one is designing that guiding region of a single guide RNA. And this is where really the science comes in. So this is kind of a baby step into the capstone project. But what they're going to do here is take the blank single guide RNA, so single guide RNA 2, add that on top of Cas9. Whoops, sorry, went the wrong way here.
So Leigh, hi.
Yeah. Yes.
I just want to make sure there's clarification for the last 2 items that were dropped in the chat prior to moving.
Yes.
So there's one question: could the strip be fed the other way?
Absolutely.
Okay, and then
and you won't get a match with this particular. We've set it up so that they won't get a match if they feed it in the other way, right? Hopefully that makes sense.
And then, so the sequence on the single guide RNA 1 would be complementary to the DNA sequence you are searching for. So that's what's in the chat.
That's right. That's right. Exactly. Yes. So, and it's specific. I don't know if you noticed here, but see how this DNA strip 1 is written upside down. This is the upside down orientation. So your students are probably going to start with this flipped over the right side up way, right? And they won't get a match when this is upside down and they're stopping at all these PAM sequences, they will not find a match on that other strand, on this strand. Okay, and so then they can flip it upside down. So that also helps them realize that, you know, DNA doesn't have a top and a bottom, right? Or enzymes don't have a top and a bottom. They're just going to scan both ways.
So does that mean that in reality, the enzyme will read it both ways? It doesn't matter which way it scanned it, it'll still target it. Okay.
That's exactly right. Yep. Yes. So, yeah, so it will, I'm not exactly sure of the physics of how this all looks. I need like would be cool. I've seen some really nice animations on YouTube. I think we have some in our playlist. I'm not sure on, I think it does do, it's looking at one strand at a time though, not both at once. It can only look at one at a time. So I'd have to scan one and then scan the other, right? Okay, so there's that. Okay, so now let's look at designing a single guide RNA. So here's our blank single guide RNA, and this is mimicking what, or modeling what scientists do when they are doing some research or trying to fix a problem, trying to change a gene, right? They need to design a single guide RNA that will bind to, or that will target a specific area of DNA. Okay, so we're going to look at how to do that. So here's our DNA strip 2. This is what they're going to use for this. And like you said, Anna, I think this is you that mentioned this. You're going to need to find, within your target region here, you would want to figure out where you want to cut, and that cut would need to be 3 nucleotides away from a PAM sequence, right? And so it's a little bit limiting in that regards. There's all kinds of ways around this. Sometimes they do 2 cuts, using 2 different systems. And so now we have the 2 fragments of DNA rather than just cut one DNA cut in half. So there's different ways around this, but the idea here is to find your target gene, figure out where your cut should be so that you can make some edits, or maybe you want to knock it out or whatever, just cut it would be fine. And that needs to be, there needs to be a PAM sequence 3 nucleotides away. Okay. So in this example, in this piece of DNA, we have given them where the cut needs to be. So imagine this is a scientist and they're like, okay, I've got this sequence and I want the cut to happen right here between this G and this T. And I've got a PAM sequence right here. We're good to go, right? So what is the guide RNA sequence that we need? Well, from the first exercise that we did, it's going to be complementary to this region right here. And so this DNA sequence, the cut site, they're just going to line up with the scissors on Cas9. That's how they line this part up. And the PAM site will line up as well. So they're going to move it along until they get that. And again, this might be upside down too, right? So when they turn this upside down, if they were to flip DNA strip 2 and try to do it upside down, they would see that the cut site, there's not a PAM sequence right here that would work, right? So they'll know it needs to be flipped over this way, okay? Once they have that all lined up, they're going to just simply fill in the nucleotides on the single guide RNA. So A, U, C, and G. And they'll get something like that. Okay. Any questions on this? And again, you can see that these are going to match this complementary strand up here, right? So they can double check their work. Okay. Any questions on part 2? This one's a pretty easy one. It does give them a nice kind of beginning look at the capstone project in this lab too. The next one doesn't have to do with the DNA model, the paper model itself, but it is looking at the specificity of different cutting things, right? So bacteria also have restriction enzymes, and those have a recognition sequence, typically of 5 or 6 nucleotides. So how would you compare the specificity of a single guide RNA recognition sequence, which is about 20 nucleotides, to the specificity of EcoR1, which is 5 nucleotides? To do that, you would want to see how many times those sites occur in the genome, okay? So this is just one way to walk through that math with your students. So we're just going to look at the human genome. So we're looking at, 3.2 billion base pairs, right? Now, to figure out the probability here, let's just start with 1 nucleotide. So A, for example, or C or T or G, it doesn't matter. It's 1 nucleotide long. It's going to occur a quarter of the time in your genome, right? About a fourth. That's not exactly right, but for simplicity's sake, we'll say, at any one location, we've got a 0.25 chance of it being an A, right? And so when you multiply that probability times the length of the genome, you get 800 million times that you have an A in your genome. That's a lot, okay? If we were to do 2 nucleotides now, so we do 1/4 times a quarter, right? So we get 0.0625. And Now we're looking at 200 million, a little bit better. Let's just say we wanted to throw in ECOR1 into our genome. How many times would it cut? Well, that is GAATTC, so 6 nucleotides. So we're going to do 1/4 to the 6, right? 0.25 to the 6. We get 2.44 times 10 to the -4. Now we're under 1 million, so we're getting better, right? But it's 1 million, still an awful lot of times that GAATTC will cut something. So the general formula here is, however many nucleotides is in your sequence in, you multiply that, it's 1/4 to the nth power. Okay. So, and then you multiply that times the length of the genome. Okay. And let's just do this for the guide RNA. So this is one of the questions in this section. It walks them through several others, but this is one of the big ones, is how many times would you expect this sequence to occur, or a 20 nucleotide sequence to occur in the genome? Okay, so we have 1/4 to the 20th, right? Because there's 20 nucleotides in that guiding region, multiply by the length of the human genome. And that gives us this 9.1 times 10 to the -13th times 3.2 billion. And that gives us this 2.9 times 10 to the -3. That's less than 1. So yeah, the odds are that you're going to have relatively few offsite targets. Offsite targets are, we wanted to cut the cystic fibrosis gene, but that 20 nucleotide sequence that we have in that cystic fibrosis gene also occurs on a totally different chromosome and messes up something else. That's what we don't want that to happen, right? And so the lower we can get that number, the better. And they can do blasts to figure out how many offsite targets there are, but having that longer guiding sequence, that 20 nucleotide guiding sequence, helps us make sure that we're not going to have a bunch of those offsite sequences. It's only going to occur once or just a few times. Okay. And if it occurs more than once, you really need to evaluate and see if that's going to be safe to use or not. Okay, so hopefully that makes sense, the specificity. Any questions on that part of the modeling activity?
So there is one question. Does the system tolerate any mismatches?
Oh, I am not the expert on that, but I have read something that indicated that it might tolerate some. I guess you could probably think of this similar to like primers with PCR, right? I don't really know the full answer to that though. But my understanding is that researchers are going to want to make it as specific as possible. The other thing that you have to remember is you also need, in addition to that 20 nucleotide region in your target DNA, that PAM sequence needs to be there as well. And so the conjunction of those is really important. I did read something about a mismatch. I'll have to look up and see if I can find that paper. Okay. But for the most part, it's pretty, high fidelity, I guess I should say. Okay. So now we figured out how CRISPR-Cas9 cuts. We figured out how to design a single guide RNA. Now let's look at how do we fix it once we've cut it, right? Cutting a gene, that may knock out a gene and that might be good, right? Depending on the situation we're trying to address. But typically what we're talking about with CRISPR is we want to cut, but we also want to repair and make a change in some way. And to do that, we're often going to need a donor template. Now I will go back on this a little bit. Remember, we had those two pathways, non-homologous end joining and homology-directed repair. Homology-directed repair is the one that uses this donor template that scientists can add along with the CRISPR system into a cell to produce the changes they want to see. Sometimes scientists just let HDR do its thing and hope for the best, right? And so that's another mechanism because most of it won't work, but sometimes there is something that will work. And so they can use that as well. But we're going to look at that HDR method, homology-directed repair, and design a donor template for that repair. And these are just here, they're not even the full thing. It's just kind of a snippet of what the the manual looks like, so you can kind of see the directions on there. Okay, so here's our cut DNA that we cut in part 1 of this modeling exercise. And on that same sheet with all the DNA, there is something called a donor template DNA little strip, and it's got a gray region in the middle. The gray region in the middle is just meant to be kind of a holding area. It's like it's non-specific repair is being made there, right? And so what they're going to do is make the homology arms. So with the donor DNA, the piece that's inserted might be, it's usually fairly long, actually. We're going to do some really short ones, but it's usually hundreds of nucleotides long. But flanking that on the outside of that change that you wanted to make, there are these things called homology arms. And that is the white region that we're looking at right here. This is one homology arm, and this is another homology arm. Those homology arms will line up with the cut DNA. And so they need to have the same sequence. And there's some fancy crossing that happens, but we've got this spelled out in the manual a little bit better. The important part here is that we just want this sequence to match this sequence, and this sequence over here on this homology arm to match this sequence. Okay, and so we're going to just real quickly copy that sequence from our cut DNA onto our donor template, onto that homology arm, and the same with the other strand. Okay. And then we're going to do that on the, where's my mouse? There we go. On the other homology arm and on the other strand of that one. Like that. Okay, so now we have our homology arms built out. And this is, again, scientists create this too, right? They need to know where their cut is. They need to design that donor template DNA to have those flanking regions that are, identical to the ends of the cut DNA, the middle ends of the cut DNA. Now we're going to introduce some changes, and the two changes in the manual that they suggest, and you can make whatever you want. One of them is introduce a frame shift. And so there's another piece of DNA that they can do all their stuff on. It's longer than it needs to be for these two examples, but you can use it for other activities as well. So if we want to introduce a frame shift, we could introduce, 1 or 2 nucleotides here. Let's just say we're going to insert a G and a C. Okay. And so that's going to be pasted into that gray region, right? So that gray region is just a placeholder, like I said. So the first thing that happens is our cut DNA will kind of bind to our donor template with those homology arms. So you would just simply move the donor DNA on top of the homology arms of the donor template, okay, since they match. And then that change is going to go in the middle. And so we're taking away that gray area, and we added our GC here. And so now we've introduced a frame shift, right? We've added 2 nucleotides, and so that's going to really mess up everything that's downstream. Any questions on that? I think the directions on this are a little bit confusing, so hopefully this helps clear it up. Let's look at one more example. So here's the same one, and now we're looking at adding an ECOR1 site. So GAA2TC, just going to add that in. Again, the homology arms are the same in this case. We're going to layer our our target DNA that's been cut on top. And then we will move our GAA2TC, that ECOR1 site, up into that gray area. And now we've added a restriction site. Okay. And because this is 6 nucleotides, we haven't necessarily introduced a frame shift mutation, but we're cutting it anyway. So in theory, that's the idea here. Okay. Any questions on the model? That is, the 4 kind of parts of the model. And like I said, you can walk through that with your students. There's also the capstone project. I'm not gonna have time to go over that today, but we do have a whole workshop on that, I think this fall. So, we'll talk more about that later, how you can use this model and kind of really go into depth and do a blast search and get some good bioinformatics data on how to design how to pick a target site and how to design the single guide RNAs. Okay. I just want to do a quick overview of the CRISPR lab. I'm not going to go into too much detail. And then what we'll do is a Q&A and the survey, and we'll be done. So we have a lab where students can actually do this in their classroom, and they are taking bacteria that is expressing, this is E. coli expressing a functional LAC-Z gene. They're plated on plates that make them turn blue, and students use CRISPR gene editing to basically add a stop codon in that LAC-Z gene, and it disables LAC-Z, so the colonies are white. Okay, the big picture look at how the process goes is that students take 2 plates, 2 different starter bacterias. One of them has repair machinery proteins turned on and the other doesn't, so they can see what happens with and without those. And 2 different plasmids. One of them has the DNA for to make the guide RNA, and the other one doesn't. So the DNA and the plasmid is transcribed into the single guide RNA, right? And one of those, the other one doesn't. They mix and match those. So they're going to have 4 different tubes that they're going to plate out on plates that contain an antibiotic to select the transformed bacteria, see what they get. And so, you know, just as a recap, we're looking for a cut and a repair. And so you need Cas9, you need a single guide RNA, you need the donor DNA, and you need the DNA repair proteins to make all that happen. So once you get all those together, you're going to get CRISPR and you'll get your white bacteria, and they've done it in the classroom, which is really cool. I'm going to stop here. I've got a survey that I'm just going to put up right quick. And actually, let me go through this right quick. The resources at the back here. So this is also in the PowerPoint I'll send out. So the lab itself is $235. It's for 8 groups. I can squeeze more out of it if I pour my plates thin, but you would need to have extra plates on hand. You could probably get about 12 groups with the amount of plasmid that we give you, and that's the limiting reagent there. If you can pour your plates thin and you have extra plates to pour them, okay? So this lab takes
the limiting factor for me is the plasmid.
Yep, the plasmid, right? Yeah. I know, me too. I'm always like, oh, I have to order more plasmid. I have so many plates, but yeah. So they give you plenty of agar. You do pour 3 different kind of plates. The 2 different kinds of plates for the starter colonies, and 1 for plating the transformants at the end. So it's 3 different types. $235. It is a lab that takes 1 full day. It really takes a good solid 50 minutes. And so if you're on a 45-minute class period, there is a stopping point and you can plate your bacteria the next day. So they can just rest at room temperature about 40 minutes in and come back and plate them the next day. Then they look at their results the next day. Now the talking about CRISPR and all that, that's not included in that, you know, class period. There's a lot of teaching that needs to go on outside of that. There's also a genotyping kit where they can look and do colony PCR, see if they made the changes that they thought they made. Did they successfully edit the LAC-Z gene? Is it still intact? And is there, you know, they have a control DNA? So it's 3 different primers multiplexed together. And so that's a really nice lab if you have a thermal cycler to do. We have this link, which has different classroom resources. It'll be in the PowerPoint, like I said. This is for all of our labs. They just redesigned the website, so it's a little, we're still learning, we're still navigating in there, but that one still goes to the classroom resources page. The manuals for this are downloadable only, and there is an answer guide that is paper that comes inside the kit. So when you get this, sorry, it's blurry. Don't throw this away. That's your answers. The manuals are downloadable. We did that because, you know, CRISPR is always changing and we need to be able to update it. So we've already updated, I think, 3 or 4 times since we launched the kit, just little things here and there, right? So this allows us to update it a lot more frequently than if it's printed. And that instructor guide is there. It shows you how to go through the capstone activity as well. The student guide is what has the models and the student protocol for the lab itself, as well as the capstone details. And here is our survey. I'm just going to leave that up, and I am ready for any questions. So feel free to unmute yourself and ask.
There is one question from Miriam. Kind of going back to the protocol, Leigh. Does the antibiotic plates not need ex-gal IPTG?
They do have XCal and IPTG. Good question. So they do. All the plates have XCal and IPTG. And for those of you who aren't familiar with that whole system, LAC-Z makes an enzyme that will break down the substrate called XCal. It's kind of an artificial lactose substrate. And when it does that, it makes a blue pigment. And it also needs an inducer in there, IPTG. So XCal and IPTG are in all three of the plates. Arabinose is in one of the starter plates. So that's what controls the expression of those repair proteins. And the plates at the end have an antibiotic, spectinomycin. That is the selection for the transformants. Yeah, great question.
So you just have to note that you cannot use regular old E. coli. You have to have the E. coli that comes with the kit because they've already been formulated.
Yes. So these are different from, say, your P-Glow E. coli, because P-Glow E. coli has a different plasmid to start off with in there. These E. coli have a kanamycin resistant plasmid so actually by the end of the transformation, you'll have bacteria that are resistant to kanamycin and spectinomycin. So the kanamycin resistant plasmid is in these E. coli from the get-go. They're also expressing Cas. They're a little bit different from the regular E. coli. So yeah, all the bacteria in this are expressing Cas, and they'll all start out blue. Any other questions? Okay, I'm going to stop recording now, and I'll still be on here for another couple of minutes. But thank you so much for coming, and I hope we'll see you at another webinar. We have, like I said, the hands-on CRISPR webinar is coming up later, and we'll have more on applications and things like that. Just go to www.biorad.com/explore events, and you can find all of our webinars. All right. Thank you very much.